So here you can see this is the DNA. Okay, the RNA is getting transcribed here. So this is the RNA, but it is called the hnRNA, heterogeneous nuclear RNA, because there is introns and exons are present, and still there is no capping done, no tailing done. So initially it is called the hnRNA. Okay, heterogeneous nuclear RNA. Now first, initially the extra part that is there present will first terminate. So it is called a terminal trimming. They terminated, they trim the terminal, the extra part. Okay, next there is a terminal addition part. It is a 5 dash UTR and 3 dash UTR. the non translated regions are added. इसमें प्रोटीन नहीं बनेगा. ओके, but they are required for the efficient translation. So initially the extra part of terminal trimming is first done, then terminal addition done, where UTR are added to the sequence. Okay, then the capping is done. First the capping is done. See, 7 methyl guanosine triphosphate cap or 7 methyl GTP cap, which is for protection, is added. Just get that's the cap. Just this part is that this come 7 methyl guanosine triphosphate comes and attaches to the UTR. Okay, see here it is attaching to the UTR, and thus the capping is done, which is for the protection. Then the cap, as soon as the capping is done, there is the tailing. Next come the 200 adenine poly A tail comes and gets attached to the 3' UTR, which is used to export from the nucleus to the cytoplasm for the transportation. So next is the tailing. So as first is the trimming of the terminal trimming, which is the extra part that is being removed. Then terminal addition, this is the UTR, so sequence of the RNA which is not included in any, it is not being translated into a protein, but they are required for the efficient translation. So this sequence are gets added. Then first done the capping. What here does the 7 methyl guanosine triphosphate comes and get attached to the 5' UTR. Next is the tail. So there are 200 to 300 adenine sequence poly A tail is mono A tail. So adenine sequence come, poly A tail comes and grant to the 3' UTR. Thus capping and tailing is done. The third step is the splicing. What happens in the splicing? The introns are removed. See here, these are the blue, the introns. So introns are gets removed and the exons gets combined together, and thus the hnRNA translated into the mRNA. Now it becomes the mRNA that is getting exported into the cytoplasm. So mRNA contains capping at 3' 5' at the poly A tail at 3' and no introns. So so this is the difference between mRNA and hnRNA. hnRNA contains introns and both introns and exons, and no capping, no tailing. But as soon as the capping is done, tailing is done, and introns are removed, only exons are present, it is called the mRNA that is get transported from nucleus to cytoplasm. See, this is the whole structure. The actual codon: 5' UTR, 3' UTR, poly A tail at 3' UTR, at 5' UTR, 7 methyl guanosine triphosphate cap is attached, which is for protection. It is for poly A tail, which is for export. This is the termination of translation, and this is for the initiation of translation. This is where the translation process initiated. So this is, I told you, UTR is not included in the translation process, but they are required for the efficient translation. So it is here where the initiation of translation begins, and 3' UTR is where the termination of translation begins, and the codons is the where the codes, this is the where the protein, this part will be the translated into the protein.